So the primary goal today is to answer your questions about the material coming up on the exam on Thursday. Remember, if you have a deadline on Thursday like the canal exam, I can make accommodation for Friday or Monday. Those are the two alternatives. But you need to contact me and set something up. If you don't have a deadline like the canal exam, the exam is on Thursday, either here at 8:30 in the morning or in the A&P lab sometime later. I can set things up for 1 o'clock, probably no later than 1 o'clock. But if you wanted to start at noon, for example, that would be okay. Okay, so, I mean, I have a boatload of, problems. They're up on Moodle that you can use for review and studying. We can go through them today. Or I can answer your questions. And of course, we can do both. Alex?
Can we go over the layers of the muscle, so like the myofibril inside of this, inside of that, how that kind of works?
Sure. Let's do that. So maybe this is a reasonable image to look at. Sarcomeres and myofibrils are inside muscle fibers, as are SR, mitochondria, all the other things that you think of as being inside a cell. This is the cell. All the things that you think of being inside the cell are inside the cell, along with the myofibrils that line up in sarcomeres. The muscle fiber, myofibrils are inside muscle fibers. Oh, it's weird. Muscle fibers form bundles that are called fascicles. The outside of a fascicle is called the perimysium. So this wrapping of a fascicle is the perimysium. The thing that wraps, it looks like it wraps fibers, but I actually think it wraps small bundles of fibers. It's called the endomysium. So endomysium is inside perimysium. Then groups of fascicles make up the muscle. And the exterior surface of the muscle is the epimysium. The epimysium is more or less the same material that forms the tendons that connects the muscle to the bone. So that's the order of anatomical structure. Yeah.
Do endomysiums wrap around the muscle fibers? So like, is that the muscle fiber wraparound?
Well, so I guess the endomysium, obviously there is a surface membrane, which is just, a phospholipid bilayer. But out around the muscle fiber is some connective tissue, some extracellular matrix. I guess let's call this a specialization of the extracellular matrix called the endomysium. And you can imagine why there is this matrix, both extracellular matrix, then some sort of connective tissue that bundles the fascicles, and then the perimysium, and then the bundle that surrounds the muscle as a whole, the epimysium. Muscles are, they're under a lot of tension. You don't want to tear them. It's a cell, but cells are fragile. By putting in this extracellular matrix, we strengthen the muscle, and so it doesn't tear under strain.
Is the endomysium to the muscle fiber what the perimysium is to the fascicle?
That's a good way to look at it. Endomysium is to the muscle fiber what perimysium is to the fascicle. So, good question. This is exactly what this class is all about. Yeah.
There were a couple of things in the concepts that I really
Didn't cover? Okay.
So, I guess,
If we didn't touch upon it in class, it's unlikely it would have very much value on an exam.
And then-
Like what? Give me an example.
Like the Cooney and Bacillus.
Right.
For second order axons.
We didn't talk about the cuneate and gracilis pathway, which are subparts of the dorsal column.
Okay, and then.
So it has a name, so in certain things, like if I didn't use those words in class, it's still true. It might actually get you value on the exam if you were to point out the cuneate and the gracilis pathways within the dorsal column medial lemniscal pathway. But, and you know, in some ways the key concepts then becomes a reference for the future, but it wouldn't be a key thing that I would probe on an exam. Is there another example you want to share?
Or like, I just like don't really understand like what each one of them is like.
Okay.
Because there were like a lot of things on the slide, and then I think we only pointed out the lateral one
Ah,
or something.
so the lateral corticospinal tract is most of the corticospinal tract, so 90% of the corticospinal tract is the lateral well, that's not quite true. So the lateral corticospinal tract is the most important part of the corticospinal tract, so that's the one you can focus on. Let's put up the slide that illustrates this. Series of slides. So the corticospinal tract starts in the cortex, as the name would suggest. The axons from those neurons in the cortex, under the gray matter on the outside and the white matter on the inside. So the cell bodies of the cortex are in the gray matter, towards the outside of the cortex. Then the axons, myelinated axons, white matter, of these cortical neurons form the corticospinal tract, also known as the pyramidal pathway, pyramidal path. Those axons go through the internal capsule, so that's part of the anatomy of the corticospinal tract. The corticospinal tract axons then go through the brainstem, and the crossover of 90% of the axons of the corticospinal tract. 90% of those axons cross over, and that crossover is in the medulla. It's called the pyramid of the medulla. So these are the things to know. So the 90% of the axons in the corticospinal tract are crossed. 10% are not. The corticospinal tract, the lateral corticospinal tract, which is the one that I asked you to learn about, is in this region of the spinal cord. It's lateral, and it's, you know, where is it? It's in the gray matter, lateral, it's in the white matter, lateral to the dorsal horn. The dorsal column is in the white matter, medial to the dorsal horn. So if you sort of, okay, dorsal column, dorsal horn, corticospinal, anterolateral, spinocerebellar, anterior and posterior spinocerebellar. If you learn those things and where they are in the spinal cord, you've got what you need. You're not focusing on these small pathways here in the anterior medial spinal. They're important, obviously. In medical school, you can't ignore them, but we're gonna focus on the most important ones here. Did that answer your question? And this is another image of the corticospinal tract crossing over in the medulla, and now in the contralateral, lateral corticospinal tract, white matter. Where do these axons make their synapses? In the ventral horn, because they're synapsing onto motor neurons. And interneurons.
Can we go over the question of the question?
Yeah. So, are we looking at question 3? No, that's visual system. You were interested in question 7? No? Just want, tell me which question. Oh, you don't have your quiz. Let me give you your quiz.
Not the last one, the one. Question 8.
Question, there is no question 8. There's question 9 twice, but do you, at least on my copy. Oh, there's question 8 and there's 2 question 9s? Okay, question 8. So, describe the anatomy of crossing over of the modalities of the somatosensory systems. The pain pathway crosses over in the spinal cord, gray matter of the spinal cord. The pain pathway makes its first synapse in the dorsal horn, and the secondary neuron crosses over and then ascends to the brain in the contralateral anterolateral. With the crossing over, a fully correct answer there was spinal cord. That's all I was looking for. It didn't give you room to say spinal cord at the level of, lumbar if it's leg and thoracic if it's, I didn't give you that information, that would be true. The pressure pathway crosses over in the medulla. If you put brain stem, I think I took off a half a point. Because it's the medulla. So in that case, just to talk through it, the neuron enters the sensory neuron carrying pressure information from, a Meissner's corpuscle or whatever neuron. The axon enters the spinal cord in the dorsal horn, does not make a synapse, ascends in the dorsal column, crosses over in the medulla, maybe makes some synapses in the medulla brainstem as well, but then makes its generally the synapse is in the thalamus. Brainstem and thalamus, crossing over in the medulla. Did that answer your question? And some of the other answers are easy too. The temperature pathway crosses over in the spinal cord. The touch pathway crosses over in the medulla. Gosh. Good. Is there anybody else here who needs a copy of the quiz so that we can be talking about it? While I'm doing this, you can be thinking about your questions, whether, quiz related or, and, anybody else? Okay. Question? I think I handed it to you as you walked in the room. Oh, no, here it is. I intended to and then I forgot. Yeah. Oh. It's up on the. Number 3. Okay, yeah, in the visual system, which of the following are correct? Let's do this together. some axons from the right retina terminate and make synapses in the right lateral geniculate nucleus, and some terminate and make synapses in the left lateral geniculate nucleus. Is that a true statement? Yes, that's a true statement, because the right retina has a right and has parts that are responding to the right visual field, and parts of the right retina are responding to the right visual field. I can see my right visual field, and I can see my left visual field. So the right retina is getting information from both the right visual field and the left visual field. The information from the right visual field goes to the left LGN. So, and the information from the left visual field of my right retina goes to the right LGN. Okay? So A is a correct statement. All axons of the optic nerve cross over in the optic chiasm. No, that's a not correct answer. Axons that are in my right retina that are receiving information from my right visual field, ganglion cells that are receiving information about my right visual field, are ending up in my left lateral geniculate, and so they cross over. Information from this part of my visual field is crossing over, but information from this part of the visual field of the left visual field of my right retina are ending up in my right LGN, and so they don't cross over. Right retina ganglion cells, axons are terminating in the right LGN. So all makes this a wrong statement. If this had been some, the answer to that would have been a true statement. Clear? Okay. So A is true and B is false. C, the axons from the left LGN, now we're talking about the LGN, so you had to read it carefully. Axons from the left lateral geniculate, terminate and make synapses in the left occipital lobe. Yes, that's true. That's the left LGN. Its axons connect to the left visual cortex. There's not a second crossing over. So C is a true statement. The right LGN receives information about the right visual field. No, the right LGN receives information about the left visual field. So D is an incorrect statement. E, axons from the lateral part of the retina closest to the left temple terminate in the right LGN. No, that's a false statement. Let me show you why that's false. Look, it's in this one. So let's go back to the question. Axons from the lateral part of the left retina. So left retina, lateral part of the left retina is here. The lateral part of the left retina is receiving information from the right visual field because of the, flipping of things by a lens. Flipping things left to right and flipping of things up and down by a lens. So the right visual field is projected onto the lateral left retina, and those axons do not cross over. The right visual field is projected onto the medial retina of the right eye, and those axons do. So if you studied this figure, you would realize that the lateral retina does not cross over. Axons from the lateral retina of both eyes do not cross over, and axons from the medial retina of both eyes do. It's another way of saying the left visual field crosses, the visual field crosses over. So this was the answer, this is the image that you had to have in your mind to answer that question correctly. So E is a false statement. F, the optic tracts contain axons of retinal ganglion cells. Yes, there is no synapse in the optic chiasm. These retinal ganglion cells' axons go through the optic chiasm, form the optic tract. So the optic nerve and the optic tract are axons of retinal ganglion cells. That's a true statement. Cortical processing of information about the right visual field starts in the left occipital lobe. Yes, that's a true statement. The right visual field information, it started to get, now if someone says, oh, wait, the LGN starts the processing, so that's a false statement. But the sentence is actually correct because it says cortical processing, and the LGN is not part of the cortex. So I'm just being a little legalistic here, but the answer is correct. Cortical processing of information about the right visual field starts in the left occipital lobe. The left visual field, left primary visual cortex. So G is a true statement. H, cortical processing of information from the upper visual field starts in the gyrus above the calcarine sulcus. No, there's a flipping there. So information about the upper visual field starts in the gyrus below the calcarine fissure. So H is false. The ventral stream of information carries information about what an object is. That's a true statement. The ventral stream projects from the occipital lobe, the primary visual cortex, into the temporal lobe, and that carries information about what it is. So this was the figure that you had to have in your mind to answer that question. Okay, did I answer your question there? So maybe while there's a gap in the action here, forming your, when you're forming your questions, let me start with maybe some of mine. So, I mean, I don't know if this is a good place to start, but why don't you work with a partner, small group, team, and see if you can put this in the order of operations that forms, this is the cross-bridge cycle of skeletal muscle. So this is the physiology of skeletal muscle. And I'll just give you a hint with this one. So the first one is D. We start with D, calcium release from the SR. In this particular scenario, there are things that happen that lead to calcium release from the SR that aren't part of this story, but this story starts with D, calcium release. So D is the first letter. See if you can work out the order of operation. I'll give you a few minutes to work with a friend and come up with a sequence of letters. While I go get a cup of coffee. next letter? D is the first letter. C is the next letter. Calcium is released from the SR, calcium binds to troponin and causes a conformational change. What's not included here is that the conformational change exposes myosin binding sites on actin. Okay, so D and C, what's next? B, I just said that, myosin binds to actin. The next one, A. ADP and phosphate unbind from myosin, and then, somebody besides Brittany? Anybody? So we've gotten to A, which is ADP and phosphate dissociate from myosin. What does that cause? Nope, not B. H. It causes myosin to undergo the conformational change called the power stroke. The conformational change from extended to flexed, which is pulling on actin. Because the myosin is bound, B has happened. No. Well, B has happened. Did we forget B? No, that's ATP, sorry. Myosin binds to actin. That's E. We've done E. Sorry, I'm getting things confused. Anyway, the next step is H, which is myosin does the power stroke. What's the next step? Oh, do you have a question?
Yes. Do ADP and phosphate unbind at the same time, or do they have differences in like between each other?
Technically, one, I think it's ADP binds, unbind. In self-is, we may have said that ADP unbinds first, and then phosphate unbinds, and it's the phosphate unbinding that triggers the power stroke.
We need to know this.
I don't think that that's an essential bit of information. So if you learn in self-is that it's ADP and then phosphate, that's probably true. Well, it is true. That's the evidence is ADP and then phosphate. But the point is they both dissociate, and that dissociation is associated with the power stroke. Okay, we've gone through H. Myosin undergoes a conformational change. What's next? Now we have, I heard the letter, B. Now we have B. B, ATP binds. What does that binding do? Next letter. G, ATP binding causes myosin to unbind from actin. Then what? F, myosin hydrolyzes that ATP that it just bound. Myosin hydrolyzes ATP. Now it's ADP and phosphate again because it's been hydrolyzed. So now we're back to myosin with ADP and phosphate binds to actin. But before that, the hydrolysis of ATP to ADP and phosphate goes to I. Myosin undergoes a conformational change to the extended state, and then now we have a cycle. We can go from I, we cycle back to E in this scenario. So the correct answer to this one, well, D, C, E, A, H, B, G, F, I. Don't remember the letters because I won't, if I give you this question on the exam, I'm certain to not use the same question, even if everything might be the same except for the letters. Don't remember the letters, remember the story, because then you can answer the question. And then I, it's not shown here because I ran out of space, but I circles back to E to form the cycle. And then we do that over and over again. We pull the actin thin filaments, we pull the Z discs together towards the M line, shortening the sarcomere, contracting the muscle. Okay, so this is a good review of the molecular mechanisms of muscle contraction, which could be on the exam. Now, this is physiology. It'll include some anatomy, but this is physiology. Let's do one more, and then I'll open the floor up for more of your questions. A physician taps hard on your knee, your left knee, the patellar tendon of your left knee. Sorry, I can't see this. Patellar tendon of the left knee. That was the right one, that doesn't matter. Draw a schematic figure, including a cross-section of the spinal cord that illustrates the sensory pathway that allows you to feel the tap and the reflex pathway that causes you to jerk your leg. So you can do this either artistically, drawing a pathway as a request, or you can use words, right? I mean, it'll be easy, some people it'll be easier to sketch it out, some people it'll be easier to word it out. Go ahead and do this problem. Both, I'll give you a few minutes to do it, sketching it out visually or sketching it out word-wise, and then we'll go through it. Remember, the patellar reflex, so if I'm holding my knee, so I can't do it, but I can feel the hit of my knee. Part of my brain is sensing that tap on my knee, and there's a spinal reflex in my lumbar spinal cord that causes me to jerk. I'm asking you to discuss, sketch, the connections, physiology and anatomy that allow that to happen. So I'll let you work on that. You can work on that in groups, in teams, whatever. I'm going to go outside and sit in that dog. Alex?
I have a question, if you want to.
Well, is it about this?
No.
Let's do this and then Is it a question about physiology?
Yeah.
Okay, let's do this and then I'll happy to answer your question. So, I'm a terrible artist, so I would not sketch this, but here are the sketches that I would try to reproduce to answer this question. So, here is the answer to question 1 in a sketch format, and here is the answer to question 2. Not sketched, but I suppose if I had to, I could sketch this out. So, but let's walk through it in words. A sensory neuron from the, and this is the right side, so the question, so this is a right knee problem. Sensory neuron from the skin and muscle tendon of the right knee, cell body in the dorsal root ganglion, enters, the axon enters the dorsal horn, and immediately, no synapse, ascends to the, in the ipsilateral dorsal column to the brainstem. This cartoon shows it making a synapse in the brainstem, not all of them do. But there's crossing over in the medulla, synapse in the thalamus. Third, so this is the first order neuron, first or second order neuron, second or third order neuron from the thalamus to the contralateral thalamus, contralateral sensory cortex. Where is the sensory cortex? In the anterior parietal lobe, the post-central gyrus. So that's the sketch of the touch pathway. And this is the sketch of the knee-jerk reflex. Stretch, axon, cell body in the dorsal root ganglion, enters the spinal cord at the dorsal horn. Here's the story that's articulated over there, so that's, right, that's what's going on here. We're not talking about that. Synapse on, excitatory synapses on ipsilateral extensors, and excitatory synapse on an inhibitory interneuron, which inhibits the contraction of the ipsilateral flexors. Stimulation of the extensors, relaxation of the flexors, jerking. So this would be the answer in a visual sketch, and then, on Moodle, this is the answer. This is what I just said in words. So on Moodle, you have the question, which you can practice and double check your knowledge, the sketch, and the description. Alex, what was your question?
I asked you earlier, I think, but I kind of forget. With regards to the muscles, we have the synapses, acetylcholine, we have acetylcholine, and then it goes into the T-tubule, right?
That's
At what
right.
level of the muscle is the T-tubule?
At what level of the muscle is the
So-
T-tubule? The T-tubule is a specialization of the surface membrane.
Okay.
So this is a specialization of the surface membrane that sort of brings these tubes of surface membrane deep into the muscle cell.
Okay. So they go to the center, so they're going to pass through all the different layers, basically.
They actually are going to surround and pass through the sarcomeres and the myofibrils.
Okay.
And yeah.
Thank you.
Can we kind of go over like the anatomy, like where the cortex is of everything in your brain? Like
Okay.
the somatosensory cortex and like the central and precentral.
Yeah, let's do that. I'm trying to think if I can find a good picture to help. Then maybe it would be best if I just draw it like you would draw it. So let's do this together.
There's one that was in like the olfactory.
Okay. try to do this with my limited artistic skills, and if necessary, if I can't do it with my limited artistic skills, I'll spend some time maybe during the break to try to find the perfect image on a PowerPoint, because I have it in my mind. I know it's there somewhere, I just can't remember which. I don't want to go through all my PowerPoints. Do you know which one it is?
Yeah, it's the chemical senses olfactory one.
Oh. Do you
I
want-
just happened
Which
to
would you
look at it.
rather do? To talk about the
Um,
image, or for me to try to sketch it out like you would have to do if I asked you to do this on an exam?
Yeah.
The latter? Okay, let's try this. Brain. What kind of view of the brain is this? Lateral? It's a lateral view, right? We're looking at the, well, yeah, lateral view. So what lobe is this? Temporal, frontal. Okay. What's that? Central sulcus. Now I'm going to take some artistic license, because we can't really see it on the lateral view, but let's just take an artistic license. What's that? The calcarine sulcus. What's this?
The lateral fissure.
The lateral fissure or lateral sulcus. Okay? Okay, so now, what is this?
Somatosensory cortex.
The precentral gyrus, which is also known as the primary motor cortex. What's that? The postcentral gyrus, also known as the primary somatosensory cortex. What's that? Primary visual cortex, above and below the calcarine sulcus. The part above the calcarine sulcus is the lower visual field, and the part below the calcarine sulcus represents the upper visual field. The left hemisphere of the cortex has information about the right visual field, and the left visual cortex has information about the right visual field. We're talking about the map. I'll stay focused on Pushka's question. Visual cortex. What's the name of this gyrus?
Superior temporal.
Superior temporal gyrus.
Gyrus.
What is it functionally?
For the auditory cortex.
The primary auditory cortex. Okay. So now it gets hard, because now where is the hippocampus? It's deep in here. It's inside here. So now let's, maybe I'll just I'll take my artistic license and make another brain. But now we've sliced off some. So we've gone into, we're not looking from the lateral, we're looking, we've sliced in. Sagittal slices, a sagittal section. So we're not on the surface, we're deeper. We can still see, in fact we probably can see better, the visual cortex. We might be able to see the central sulcus and the precentral gyrus, motor cortex, and the postcentral gyrus, the somatosensory cortex. And now we're getting into the medial temporal lobe, and we've cut away the auditory cortex, the superior temporal gyrus. It's out here somewhere. And in here, medial temporal gyrus. Not midline, because we haven't gone all the way to the midline. We're in the middle of the left hemisphere. There's going to be the olfactory, like I said, bad artist. Olfactory cortex is there. Hippocampus is in there somewhere. And the taste cortex, just run out of color. The taste cortex is at the junction between the frontal, parietal, and temporal lobes. It's called the insula. Remember, all these things are not physically separated. We give them names, but they all are connected. So the fold that connects frontal parietal with temporal in here, in this deep section, it's called the insula. That fold is where the taste cortex is. I didn't teach that, so I'm not going to test you on it. But that's the relationship between the taste cortex and the olfactory cortex. So let's just see if we did this right. Which PowerPoint did you say it was in?
Like chemicals include olfactory and stuff. Or
Chemical
olfactory
senses,
and
right.
this one.
Got it. Smell and taste. Is it all the way at the bottom?
It should be like before we started talking about these. So it was the last one.
Oh, go to Whole load of clickers on it. Auditory cortex, pulling away, going deeper, the olfactory cortex, and the taste cortex on that fold between frontal, parietal. It shows it being frontal, but it's frontal, parietal, and the fold with the temporal lobe, taste cortex. I'm not asking you to learn about association areas, but ventral visual stream, what it is, dorsal visual stream, where it is or how it's moving. So this has more information than I've asked you to know, but it also has some information that I have asked you to know. Let's take a break, and you can think of your other questions. Maybe what I'll do is skip a bunch of the stuff that I have on my review PowerPoint and go to some clickers. We'll see. Let's take a short break, a couple minutes, and then talk some more about your questions. The exam will be focused more on physiology than on anatomy. And so we should probably, even though much of our discussion here has been about anatomy, but you know, function determines structure, and structure determines function. Anatomy informs physiology, physiology informs anatomy, so it's not, the time we've spent has not been wasted, but let's make sure we get at physiology too, because that, the exam will. Aiden asked about taste physiology. What are things to know? You need to know the 5 taste modalities: sweet, salty, umami, bitter, and sour. That's physiology, that's how taste works. You need to know that there is a taste bud. There are taste buds on the tongue. I don't care that you know about the different names of the papillae of the tongue. That's anatomy I didn't emphasize. It's not on the key concepts list, I don't think. But there is a taste bud. The taste receptor cell is a short receptor cell. So it responds to a chemical stimulus and then stimulates the afferent nerve. So that's physiology. And this is the key physiology of taste. Sweet bitter, and umami activate GPCRs, different GPCRs. Obviously, the sweet GPCR is not the same as the bitter GPCR, but they are both GPCRs. And so is the umami bitter GPCR. Different GPCRs for the different taste modalities, but they're all GPCRs, and they all couple to the GQ story. I can walk through the GQ story, but I've done that a lot. Maybe in the interest of time, I'll just say, you should be able to tell me everything you can about the GQ story if I ask you about taste receptor physiology. That's the story of these three modalities. Increasing calcium, right there. By the time we get to increasing calcium in this receptor cell is going to increase neurotransmitter release from this synapse. I could ask you to tell me about neurotransmitter release. That's going back to the first exam. Calcium binding to synaptotagmin, snare, conformational changes, fusion, release of a neurotransmitter. What neurotransmitter actually is unknown? So I wouldn't take, if you can't name a neurotransmitter here, that's fine, because I couldn't. But excitatory neurotransmission, depolarization, action potentials in the afferent taste neuron. So for GPCR, for taste, for bitter, umami, and sweet, you've got to know the GQ story. For salt, you simply have to know that it's a sodium channel. Sodium influx, salt is sodium chloride, sodium influx, depolarization, voltage-gated calcium channels, calcium storage. And for sour, you have to know that it is a channel mechanism, but it's not a proton channel, it's a proton block of a potassium channel. Proton block of a potassium channel, block of potassium efflux, depolarization, calcium influx, neurotransmitter release, Okay, so that's the key physiology of taste. It's summarized in this slide too. Calcium, neurotransmitter release. Here it's called serotonin. That's one of the neurotransmitters. There's about several, I think. But if you can narrate the stories on this slide, you've got taste physiology. Other questions about this, physiology? Yeah.
If there is something you think that we absolutely need to go over, or you think will be super important that we haven't covered regarding physiology?
Oh, it's all absolutely important.
Yeah, I know, but
I can't answer that. I don't know the answer to that question.
okay, let's look at olfactories.
Okay, let's look at olfactory. We have more than 5 modalities of olfactory, way more than 5, like 10,000. They all interact, all of them start with an activation of a GPCR. Simple. But it is not GQ. It is GOLF, which there's a story here which you can tell me and should be able to tell me if I ask you about olfactory physiology. Adenyl cyclase. There's a story here that you know that you should be able to tell me. Right? And this is clearly this is related to the GS story, but it is not GS, it's GOLF. Cyclic AMP. But we're not activating pKa here, which is the canonical thing that cyclic AMP activates. We're activating a cyclic AMP-gated cation channel. So olfactory neurons express this cyclic AMP-gated cation channel that other cells don't. So this story leads to sodium influx, depolarization, remember, olfactory neurons are olfactory receptors are long receptor cells, so they have an axon that projects to the olfactory bulb. There's anatomy, but if you remember that anatomy, you'll remember, oh, I don't have to just go to calcium and neurotransmitter release. I've got to include action potentials here because it is a long receptor cell. So being able to say, okay, depolarization, threshold, opening of voltage-gated sodium channels, da-da-da-da-da, depolarization, the action potential story, and then get to the neurotransmitter release in the olfactory bulb. So that's anatomy informing, reminding you some physiology you've got to tell.
Yeah, that's a good question.
So, binding, GPCR story, adenylyl cyclase story, cyclic AMP story, cyclic AMP gated cation channel story, depolarization, action potential story, neurotransmitter release story, olfactory bulb, processing of the information in the olfactory bulb and then in the olfactory cortex. These axons go to the olfactory, this is the olfactory bulb, these axons go to the olfactory cortex. Aiden?
This might be a really stupid question, but out of the sort of the sensors that we've looked at so far, which ones have short neurons?
Okay, let's do this. Which sensory modalities have short axons? Are short receptors? Which ones are short receptors? Name one. Taste. We just did that one. Vision. The retinal photoreceptor cell is a short receptor cell. Hearing, auditory. The auditory hair cell is a short receptor cell. That's it. Which ones are long? Touch. Well, so somatosensory, which includes touch, temperature, pain, pressure, proprioception. All of that is somatosensory, and all of those are long receptor cells, and olfactory. They're not very long, but they're considered long because they have axons. So that's anatomy, sort of, but that's also physiology. So this is the key slide for understanding the molecular mechanisms of olfaction. This is the key slide for knowing sort of the molecular anatomy of olfaction and being able to combine anatomy with physiology. And we already went through this slide with olfactory cortex anatomy. And this slide is just getting at the idea that we use combinatorial activation of different receptors in order to differentiate related smells. The key word to this is combination. Combination of activation of different receptor cells gives us our ability to differentiate 10,000 odorants as opposed to just 5 cases.
Do we know like the number of receptors that dogs have versus humans versus like that kind of detail?
Absolutely. Can you memorize these numbers? No. Yes?
Can we kind of go over like the T-tubule stuff for the muscle?
Sure. So I wish I had a model because this is a 3-dimensional structure. Is this a slide that would help?
Yeah, really my question was just like with the
Ryanodine?
ryanodine receptor, is that just the little, I know we kind of went over this in lecture, but is that just the little thing that connects like the DHP to the calcium
Yeah.
receptor? Okay.
Yeah, well, so the ryanodine receptor is the calcium release channel from the SR.
Okay.
And it does connect the DHP receptor in the T-tubule with the SR membrane. And so when it opens, when the ryanodine receptor calcium release channel opens, calcium is released from the SR, calcium binds to troponin. Now we're telling the story that sequence, that we don't have to do that again.
Okay.
So the voltage sensor, or DHP receptor, is in the T-tubule membrane, and the ryanodine receptor, calcium release channel, is in the SR membrane. This is very close. They are physically, in skeletal muscle, these are physically connected to each other. So a conformational change, a voltage-dependent conformational change of this voltage sensor in the T-tubule. Remember the action potential from the surface membrane. T-tubule is a specialization connection of the surface membrane. So action potentials on the surface, you should know how that happened. Action potentials on the surface membrane penetrate into the T-tubule. Conformational change, opening, calcium release, binding, conformational change. You should be able to narrate this story too. That's the molecular mechanisms of calcium-dependent activation of contraction. This is the upstream story from that letters problem where it led to calcium release.
Thank you.
So I was coy about how we get action potentials, but this is the story you could narrate for the physiology of how we get action potentials in a muscle cell. Let me just sort of give you the key words. Action potential in the motor neuron. Calcium channels. Calcium influx. Synaptotag. Snare. Conformational change. Fusion. Release of acetylcholine. That's a key that we know that's a key neurotransmitter. Acetylcholine. Acetylcholine receptors. Sodium influx. Depolarization. Activation of voltage-gated sodium channels. Activation of voltage-gated sodium channels. Action potential. Propagation to the T-tube. If you can narrate that story, you've got the molecular mechanism for how a motor neuron causes an action potential in a muscle cell. And then if you can narrate the story that I told before, you can narrate the molecular mechanisms for how an action potential causes calcium release. And if you can do the letter problem that we started with, a few minutes ago, half an hour ago, then you're telling the story of how calcium causes contraction. So all of those are important stories to be able to tell about muscle physiology. Alex?
Which neurotransmitters do we know for the sensory? Like, we know vision, we know this one.
We know vision. Glutamate is the neurotransmitter released by the retinal photoreceptor. We know that acetylcholine is the neurotransmitter of the motor neurons. We probably know more than I'm indicating, but those are the key ones. Let's see. The neurotransmitter released by the auditory hair cells, I'm not sure I know, so you don't have to know. The neurotransmitter released by sensory afferents, the primary afferents of the sensory system, probably glutamate, but I don't know, so you don't have to know. So I don't want to muddy things. Glutamate at the retina and acetylcholine at the neuromuscular junction. Those are key.
Can we go over the sensory physiology? Because we had there's the different capsules, whatever they're called, that we have. It was one of the first lectures, but it was the physiology of when you perceive touch.
The physiology of perception of touch. So, you mean all those corpuscles?
Yeah,
It's probably not worth spending time on, because I'm not asking you to know the names of the different corpuscles.
Okay.
I'm just asking you to know that there are corpuscles that provide information about touch, pressure, and sort of vibration.
Mm-hmm.
And then there are free nerve endings that provide information about temperature and pain.
Okay.
So they're not corpuscles, they're just sort of nerve endings.
But we don't need to know the names of them.
You don't need to know the names of the corpuscles. So in retinal physiology, So, you know, let's see, haven't done vision in a while. That's how, you know, what are some of the visual system physiology? We're got a whole bunch of clickers. But photons bind to retinal, rhodopsin or opsin. This is key physiology. This is, you should be able to narrate this story of how a photon of light is converted into a depolarization, a hyperpolarization. How dark leads to a depolarized retinal photoreceptor cell, and light leads to a hyperpolarized retinal photoreceptor cell. That's a molecular physiology to know. I'll do it if you want, but this is the slide that you should be able to narrate.
So if it's hyperpolarized with light,
Light causes hyperpolarization.
so what is this thing triggering around?
It's going to trigger a decrease in, well, it's going to trigger a decrease in neurotransmitter release. And that decrease in neurotransmitter release from the retinal photoreceptor cell is going to be converted, depending on the circuitry that it's connected to an increase in action potential frequency of the retinal ganglion cell, or a decrease in, or an increase or a decrease in the action potential frequency. It's the ganglion cell that fires action potentials. And there's a circuit between the retinal photoreceptor, the bipolar cell, and the ganglion cell that tells the story of how a hyperpolarization of the retinal photoreceptor cell can either cause a decrease in action potential frequencies or an increase in action potential frequencies in the retinal ganglion cell to which that retinal photoreceptor cell is connected by a bipolar cell. That's a story. The on-center and off-center story is physiology to know. We spent, I don't know, 20 minutes in class going through it. So what's your strategy for studying for this test? I would recommend using the clickers that are available. They're embedded in certain PowerPoints, but also I think the review session, the PowerPoint that I prepared for today has scores of clickers that I probably pulled from the ones that we didn't do in class. So you could, and the clicker is there, and the answers are there. The short answer, like that, you know, the sketch of the somatosensory system, or the sketch of the knee-jerk reflex, and the story is on that PowerPoint. So you can start with the PowerPoint and going through it and identify where your strengths are and where your weaknesses are. My recommendation is, you know, be able to outline all of these molecular stories from scratch. Go back and then check your knowledge against the PowerPoint or the textbook, and then fill in the gaps. You know, if you, oh, I forgot GTP. I got to remember to write GTP binding to, you know, if it's a G protein thing. What's going on? A baby? I get it. Yeah, I would have guessed. That light up is baby. I'm happy to answer questions between now and Thursday. Remember, if you take the exam on Thursday, remember that there are people who are not taking it until Friday and Monday, so please don't talk about the exam with people who haven't taken it. Some of you I will see you in lab this afternoon.